All right. Can you hear me? Yeah. Good. Let's get started, guys. We're almost done. Almost done the semester. 3 more topics. Well, 2.5, and then that's pretty much it. So I think it's time for you guys to start studying. Well, not really, because the final is on the 23rd, right? Who has a holiday during the week of the exam? Okay. Anybody else? Okay, so guys, send me an email, all 4 of you. Ilan, and Gabby, right? Okay, cool. The rest I got it. Okay, so I'll see what I'll do with that. Probably gonna have to make an alternate exam or something like this to avoid differing the grade, right? If there's anybody else who's not here today, but who's watching the YouTube, also send me an email so that I can take care of that, because I need to know how many students will be there during the final exam. Also, like, share, and okay, no, seriously, guys. So today we're going to cover regulation of gene expression, because what happened during the beginning of the semester is that we were studying genes as being either there or not there, and that affecting the phenotype. But actually, this is not how it works. There's many layers beyond just having the gene that dictate if the phenotype is there or not. And so today we're going to start covering that. And we're going to do that by looking at the lac operon. The lac operon is quite simple. It's one of the simplest pathways that we have. You guys can't see there? One second. Yeah. Just 1 second, guys. Okay, let's just wait a second, and then we'll continue. There's always something messed up with this thing here. Every time I come, I have to fix it. So today we're gonna. Okay. I guess not. Okay. Is it okay if you guys look here? You're okay? You're good? Okay, so I guess we'll do it this way. I'll figure it out after the lecture. What did I say? So I was talking about this lac operon here. So when you're going to go through your biology program or biochemistry and whatever, you will probably see a lot of pathways. And so it's very important to understand this basic one so that in the future, when you see like a huge network, you don't get scared, okay? So these guys, Jacob, Mono, and I forgot the other guy, what's his name, but they were studying this lac operon, and they made a lot of contributions in terms of genetics and gene regulation. And one of their famous quotes is, anything found to be true of E. coli must also be true for elephants. So the basics are always the same. It's just, in eukaryotes and all that, it gets a little bit complicated. The reason why I called this slide the OGs, for those that know, this guy is smoking, this guy is playing dice, so it's not looking really like they're doing science there, but good PR for them, okay? So gene regulation, there's 2 types, okay? Very simple. Either it's on or off, right? So there is positive regulation or negative regulation. And usually it happens like this. So let's take a look first at positive regulation, and then we'll take a look at negative regulation, right? So the idea behind your DNA and then transcription and translation is to produce a protein that will have a certain function, okay? And that will give you your phenotype downstream. So there are 2 ways to decide if you are going to express that protein, that gene or not. And this is through these 2 mechanisms. The first one being the positive regulation, where you have either, like we saw last lecture, post-translational modifications that will make a protein more active or whatever. Or you also have these proteins that are called activators which increase the efficiency of the RNA polymerase, for example. And what this will do is that it will bind better to the promoter and transcribe more efficiently the gene. Yeah. So there's a lot of mechanisms. It's not just one thing. I mean, in this case here, we're going to look at allowing and improving, both of them. But just imagine it like this. Any mechanism that makes more protein, right? Anything that promotes more protein is a positive regulation mechanism. Obviously, you can make it bind better so that it's more efficient at walking on the DNA. What else can you do? You can also make it transcribe faster, things like this. Okay, so this is positive regulation. Usually not usually, all the time the RNA polymerase will bind to the promoter, right? And there is usually an activator site that is upstream of that, where an activator would bind and promote transcription. Okay, so that's that. So if you have this activator will just activate transcription. In this case here, if you don't have it, there's no transcription. Okay. The other side, the negative regulation, usually instead of the active site, you will have an operator site where a protein called a repressor will bind to the operator and prevent transcription. And this is usually done structurally. So what happens is that the RNA polymerase will bind to the promoter but cannot go through the repressor and will just get off the DNA. And so if there's no repressor, obviously, then you have transcription happening. Then you can imagine how both of them can interact together at the same time, and it's the net of repressor and activator that decides if you're expressing a gene or not. Cool. One thing that was recently discovered in terms of gene regulation and expression is called epigenetics. So you have the genes, but then there's a higher layer of control, which is mostly affected by the environment, food you eat, etc. So just the exposure that you have with your environment is actually affecting the regulation of certain genes. Okay. One domain where this is studied a lot is aging. So there's aging has a epigenetic component, and you can read on this. It's pretty interesting. Now, these proteins that act as repressors or activators or enhancers or whatever, they usually have a DNA binding site, and they also have another site where a certain molecule can interact with it, right? In terms of the activator, when you have this molecule called an effector bind to it, it will allow it to do its job of an activator. Okay, so it activates the activator kind of. Okay, this site here is called the allosteric site. You probably know this from CJEP, where the effector will come and bind and change the structure of this protein to allow it to do its job. Same thing for the repressor, but you just have to think in the opposite sense. So if the effector binds there, it will inhibit the inhibition. So it will kind of activate the gene transcription. Questions up to now? No? Okay. So the lac operon. Did you guys already see this before? Probably. Yeah. Okay. So we're going to review it a little bit more in detail, but not that much. Just have to remember the terms that I'm saying. Okay. Today I'm trying a different type of lecture. As you can see, there's not a lot of text. I put just enough text. So I'll be explaining the diagrams here. So try to write at the same time. See how this one does here. Okay. I'm still practicing my stuff too. Okay. So what did I want to say? So the lac operon is composed of two segments. The first one is the I gene, this gene here, which is a gene that codes for an inhibitor, which serves as a repressor of the lac operon. So when this protein here is made, it will prevent the transcription of the lac operon. This section here is called a polycistronic region because it has many genes in it. Remember, bacteria, the mRNA usually has not only one gene, but can have 3 to 4, 5 genes, okay? So this is a polycistronic sequence, and it encodes 3 genes. beta-galactosidase, permease, and transacetylase. These two are very important. This one we don't care about for this course. Actually, we almost never care about it unless you're doing research about it. But it's just a protein that will change some sort of bond in the sugar. Okay, so forget about this part here. The A, we don't need to know it. You still need to know the structure, but not the function of the A. It's not important. Okay, what's important is the other two proteins, the beta-galactosidase, which is an enzyme that breaks a sugar into two, and permease. The name says it, permeates, so it allows for. So this is kind of a channel that allows for lactose, which we'll see in a second, to enter the cells. Okay. Upstream of this polycistronic region, there is an operator sequence, which is right here, and a promoter sequence. So repressor binds to the O region, RNA polymerase binds to the P region, and then they encode an mRNA that contains 3 genes, Z, Y, A, which are these here. This one here breaks a sugar, this one here allows a sugar to get into the cell, and this one we don't care. Okay. So one thing that's important to know is that certain genes are trans-acting and other genes are cis-acting. So the repressor, because it's coded in a different region, it will be trans-acting. That means that it will act on a different DNA strand. Okay, it will come from one place and act on another one. Okay, so that's trans-acting. And this will be important to understand when we talk about diploids in this sense. Okay. Let me just put this up. Okay. The other ones are all cis-acting. So O is cis-acting. Okay. Make sense? Because O can only block the genes that are right after it. Right. So if there's an inhibitor that's bound here, then this sequence here is cis-acting in relation to these guys. So what I mean by cis and trans-acting is just where the effect of, let's say, a mutation on this one, for example, happens. Okay. For example, in here, the I, right, it makes a protein that floats inside the cell and can come and bind here. So that's trans-acting. It's going from here to there, right? But then this sequence here, if there's a mutation here, it does not affect this side here. It only affects everything on the same strand, right, on the same DNA sequence, and so that's called cis-acting. And you'll see in a second what that means when we get into the details. But keep in mind these two terminologies. This repressor is trans-acting. Yeah, because this one makes a protein, but this is not a protein here. This is a sequence. Exactly. This product here, the repressor, is trans-acting because it acts on a sequence far away from it. But this one here is just a sequence where something binds. And so this one here, its effect on the operon is cis and not trans. Okay. Yep. All of it. I-P-O-Z-Y-A. This is all the operon. I mean, yeah, the whole thing. Just consider it the whole thing. Yeah. I am not sure exactly what separates it, but it's been a long time I didn't look at this, but I think this is on the chromosome of the bacteria and this is on a plasmid, or one of the plasmids is in there. I have to make sure for that. I'm not sure. Either they're on the same bacterial chromosome, so the only chromosome that, let's say, E. coli has, or this is on a plasmid, okay, or some sort of DNA sequence is in there. All three are possible. Most probably they're on the same chromosome, but it's not that important. Okay. So let's start by the Z gene. So beta-galactosidase. So all you need to know about this is that this enzyme will break a beta-galactoside linkage, which is between so between the two rings of the lactose, and it will generate galactose and glucose. So basically, lactose is just glucose attached to galactose. Okay, lactose can also be altered to allolactose, but we don't care about this. What's important to know is just this effect here. Okay, so when you give the cell lactose, it will make glucose and galactose, and then use that glucose. Okay, this is the stuff you've probably seen. Correction, the operon, whoever just asked me, just consider it as this, okay? I don't want to mix you up. So the operon is posi YA, okay? And this one here, consider it as outside of the operon. So the idea here is when we're looking at the lac operon, we're trying to figure out if there is a transcription of the structural genes or not. And that's pretty much it. And is the regulation negative or positive? And we'll take a look at different scenarios where that happens, so you can see how this happens. So initially, when there is no lactose present in the cell, right? The cell will be able to, so it will make this inhibitor here, this repressor protein, which is the I, the inhibitor. It will make the mRNA, the polypeptide, and then fold it into the repressor protein. This repressor protein, because it is not bound by an effector, it will go to the operator site and prevent the RNA polymerase from walking on the genes and making these proteins. Okay, so it will come up here, and it will structurally prevent RNA polymerase from making the transcript, which contains the 3 structural genes. Okay. In the case of lactose, when lactose is present, this is a bit different. Here, what you have is that the repressor protein is still made, but lactose acts as an effector, right? And so it will bind to the repressor and change its structure and prevent it from binding to the oocyte. And so what this does is that RNA polymerase does not have anything blocking it and will be walking on the DNA to make the mRNA with all these 3 genes, followed by the translation of these proteins. Cool? Yep. The lactose is kind of floating, and then whenever it comes into contact with the repressor protein, it binds to it. Yeah. I mean, in reality, it's not like you have lactose, it automatically binds and goes there. So what happens is that like you imagine all of it moving at the same time, okay? If there's no lactose bound to the repressor protein, it will still bind here, but then it will get removed. And so it's kind of a dynamic, right? In the case here where there's no lactose present, the 2 options are not available, right? This one will block the transcription of these genes. But here, because you have 2 forms of the repressor protein, then sometimes this one will bind by itself, and sometimes this one will bind by itself. See what I mean? So if you put a lot of lactose, this is like unrelated to the course, okay? But just to give you an image, if I put a lot of lactose in the media, there's a lot of lactose molecules, so a higher chance of them interacting with the repressor. And so there will be a stronger inhibition on the inhibitor. But if I put a small dose of lactose, what's going to happen is that there will be only a little bit of lactose to prevent this inhibitor from doing its job. And so most of it will still be repressive. Okay? But we are studying it as either or. Okay? This way it makes it simpler for us. Yes, Joe? In this case, there are mutations where the I is not made. I will show that in a second. Pedro? In this case here, no. It's only lactose. But there is a synthetic molecule, which I will talk about in a second, that's used to study this. Because what happens here, when you're adding lactose, beta-galactosidase can break it. And because it can break it, it's very hard for you to study it. Because you put lactose, and then you start making this here, and beta-galactosidase breaks it, and so you lose the lactose that's supposed to inhibit here. So there's another molecule that was made synthetically to do these studies, and I'll show you that in a second. Sign. Yeah. Degraded. Yeah, it's exactly that. Because the idea behind this, like the cell is very smart. If you think about it like this, the cell imagine a cell as being like a human there. It sees lactose, it's like, oh, there's lactose. Now let's take care of lactose. And so lactose kind of inhibits, it's the inhibitor of the operon, and then it starts degrading lactose because lactose is present, right? So this is a mechanism for the bacteria to detect lactose and use it for energy. Make sense? Yep, exactly. Same thing that happens for many things. For example, for humans, if you eat meat, you will activate pathways in your body that will try to break it down into amino acids, and then use to rebuild muscle or whatever. Your body is not going to start like activating lipid enzymes when you have, when you're eating meat, right? So there's detection mechanisms like this. This is the one for the presence of lactose in the medium for bacteria. Easy? Okay. Let's complicate things a little bit. So the first thing that we have to look at, so there are variations of these genes. Remember we were talking at the beginning of the semester about there are different alleles for the same gene. So if we look at the operator site, there are multiple different variations of this gene. One of them is called the OC, and this is the one that we're most interested in. There are others, but we will just focus on 3 of them: O-, OC, and O+. Okay? O- means that so not this one here, this is plus, but just telling you, O- means that the operator sequence cannot be bound by the repressor. Okay? OC is the same thing. It cannot be bound by the repressor. And why is it called C? It's because it's constitutively active. Okay? Hard word to say. But what it means is that it's always active because the repressor cannot bind it. So this could be just, for example, a change in few nucleotides that make it so that the repressor does not recognize this sequence anymore. Okay? So, yeah. Yeah, on these genes here. Yeah. So then when you have I+, that means you're making the repressor, but because this sequence here is OC, then the repressor will not be able to bind it, and you will be expressing these genes. In this case, you don't need lactose to express the genes because the promoter has no way of being inhibited. This what we were thinking earlier in terms of like just haploids, right? Just one second. Now we have to think also about diploids. So scientists, what they like to do is take 2 bacteria, put them together, conjugation, to study different proteins. In this case, when you see this notation here like this, it means that we're talking about a diploid, right? And you can see here that this I+ here can be made either from this strain or this strain here and act on both genetic materials. Does that make sense? So 2 bacteria come in, one gives this DNA sequence, the other one gives this DNA sequence, and so the repressor will be made by both, and it will act on both operons. Assuming this one here, for example, is I-. So in that case, this bacteria here does not make the repressor, but because it's a heterozygous bacterium, this is sufficient to come and act here. This is what I mean by transacting. Okay? Fine. In this one here. Yeah. Right. So it's pretty much useless. It's just it's a mutation that messes up the whole thing. That's the idea behind it, right? And this is how gene expression is kind of regulated by different mutations making different alleles of the same gene. Okay. In this case here, it's just a mutation that has no point because it turns the bacterium into a bacterium that cannot sense lactose. Right? Yep. Same thing. Exact. Yeah. Usually OC is like a deletion or something like that. But OC, there are specific mutations that are substitutions. Okay. Minus usually means you don't have it, right? Or most of the time. Easy? Okay. Let's look at the table, which looks like the exam. Okay. So, as I told you a few minutes ago, we don't use lactose to study these kinds of systems because it will be degraded. So there's a molecule that is called IPTG, which is synthetic, and this one here does not get broken down by beta-galactosidase, but still acts as this little guy here. Okay. And so if you are given a genotype like this, you have to be able to kind of determine what's happening, and then but with the addition of IPTG, do you make this protein here or not? Okay. So let's say, for example, we have a bacterium that is, so all these strains are I+, as you can see here. And imagine we start with the first one here. Let's say you have this strain here, which is O+, Z+, Y+. So then if you add IPTG, right, IPTG is going to act on the inhibitor and prevent it from inhibiting this operon. And so when you add IPTG, you will be making Z and you will be making Y. If you don't add IPTG, then the inhibitor will be able to bind to the operator site and it will inhibit the transcription of Z and Y. Make sense? Yeah? Okay. Second one. When you see it like this, this means it's a diploid. Okay? So in this case here, we have the only difference that we have here is that first it's a diploid, and then you have Z+ here and Z- here. Okay? So we have to think about it in a different way. Okay? If you get these like this, the easiest way for you to do it on the exam or a quiz is to actually just draw it, because then you can see what's bound where and then what's working and what's not working. Okay? So if I have the I+, let's look at this operon first. It can still bind to the operator. And if we look at this operon here, it will still bind to the operator. So then we need to induce it to remove it from there. Okay? If we induce it, that means IPTG will bind to this inhibitor here, the I+, prevent it from binding to here, and prevent it from binding to here. Make sense? So then, will you be making Z, beta-galactosidase? You will. You will be making it from this one here, but not this one here, right? But the general end goal and the result is that you'll be making beta-galactosidase, because this one is sufficient to replace the function of this. Okay? And why? Well, they're both wild types, so then that means you'll be making it when you induce it. Okay? This is what it means. Z is dominant to Z-. If you have it in a diploid state, just one of them is sufficient to produce this. Okay? Yeah. Yeah. If we had, the enzyme. So just one copy is good enough. Okay. If we had both of them Z-, then you would have a minus sign here. Because yes, you are inducing it, but the gene does not make a proper protein. Okay. Now the third one is where we have this constitutive operator. And in this case, I+ cannot bind to the operator. And so induced or not doesn't matter. You'll still make them. Okay. Both enzymes. Now it's important to think before you answer this fast. When you see OC, right, don't automatically think, oh, it's already, it will never be inhibited, so I'll just put plus, plus, plus. You could make the mistake of like, it's OC, Z-, Y-, and then, right, just because you were jumping to conclusion with the OC, you forgot about these two, okay? So in this case, it works. But let's say you had OC Z minus Y minus, then all this here would be minus. Okay. Cool. Last one. Yep. Because the gene to make that enzyme is mutated, and so you don't make the enzyme if it's Z minus or Y minus. Nothing. It's very much useless. Induced means that you're adding this IPTG, which is kind of the lactose, similar to lactose. Non-induced, that means you're not adding the IPTG. Yeah. Sorry? Yeah. Operator. It just means that it's always active. Or always something, either active or, right? So just consider it as being the operon is always active. It can never be inhibited. That's why it's constitutively expressed. Okay. Should I draw this one here with the Z minus and Y minus? Yeah? Okay. So where's the... Just 1 second, guys. Okay, so we had this one, right? OC Z plus Y plus. So when you draw this operon here, it basically looks like this is Z, and then this is Y, and you have your OC here. Okay. So here there's nothing that can bind. Because this is Z plus, then you're able to make this protein, and you're able to make this protein. Okay, simple. If we remove this, right, and we make it, you know what? So if you see something like this, that means that the sequence here is just messed up, okay? So it does not make anything. It does not make anything, okay? Now, if this one was Z minus and this was Y plus, then this sequence here is messed up, but you still have the Y plus. And so that will be transcribed. Easy? Okay. All right, last one, this one here. So, geez, have to go faster. In this diploid here, what we have is one operator that is plus and the other that is OC. So we're making I plus already. All strains are making I plus. I plus will be able, the inhibitor will be able to bind to the O plus, but not to the OC. Okay, so in the case of beta-galactosidase will not be made in this case here, but will be made in this case here. So that means that induced or not, you will be making beta-galactosidase because there's an OC here. Okay. Now for the permease, gene Y, gene Y is not made in this sequence here, right? And so when the inhibitor comes, binds here, transcription happens, but because you have Y minus, you're not making Y. So then we have to look at the other one. I comes and binds here, and then it inhibits, right? So if we don't induce it, we're not going to make anything here. If we induce it with IPTG, we'll be making the Y. Good? Yeah. F is the F prime. It's just, remember in the bacterial conjugation when I was talking, right? So this is just to show you that one of them is HFR, the other F, right? But it's not important in this case, okay? Okay, more of this. So LAC-I has different variations. Again, LAC-I can be either negative, or is it the next one? Or it can be IS, Okay, let's start by I minus. If you have I minus, that means it does not produce the repressor. I plus means you produce the repressor. So in the case of a diploid like this, just one copy will be able to act on both operons. Okay, simple. Yep. So if we have a diploid like this, where one of them has I minus and the other has I plus, just because this one is producing the inhibitor, it's sufficient because this inhibitor can travel inside the cell and go and bind to both operators. Exact. In a diploid, obviously. Let me see if we can take one here. This is, so if we take a look at this guy here, I minus Z plus Y plus, because we don't have the inhibitor here, doesn't matter, we'll be transcribing these genes non-stop because there's no way to inhibit this operator, this operon, okay? If we have a diploid though, one of them being I minus and the other one being I plus, then it's a bit different. This I plus is sufficient to act on both operators, okay? And then you can do the thinking about this, okay? Just, yep. No, forget the crossover. Just when you get something like this, draw it like this and then see where, if there is a repressor, where it can bind and where it cannot bind, and then you'll have an idea about the rest. Okay, yeah. Exactly. So if you, this one here is basically just not this, it's drawing this one here. Okay, and then because there's no I, there's nothing that binds here, and so you end up making this non-stop. Okay, yep. It will be, I will tell you. Here it's written that it's O plus, right? But sometimes it will be OC, sometimes O minus. Even the promoter, sometimes it will be P plus, sometimes P minus. So think about that, okay? Yep. Yeah. How do you make the conclusions? So there's only one type of question really I can ask, is if I induce, do you make this gene or not? Or that's pretty much it. Kind of something like this, right? Or if I, do I need IPTG to make these genes, or can I make it without IPTG? Something like this. And so what you do is you draw these. If there's a repressor, then you have two scenarios. The repressor being able to bind, or the IPTG coming and binding to the repressor and preventing it from binding here. Okay? So that's what I mean by induced and not induced. Cool? Where is that? Constitutive. It means that in this sense here, I is negative, and so it does not inhibit the operon, and so the operon is expressed constitutively, which means without any inhibition. Okay? Okay, more complexity. So there's another variation of the inhibitor gene. It's called IS. So then IS is dominant to I plus, which is dominant to I minus. Okay? So the difference is very simple. When you have the IS, it cannot be induced. It's a super strong inhibitor, and so the IPTG or lactose does not work anymore. Okay? So when you have IS, it doesn't care if there is induction or no induction. It will be inhibited no matter what. Okay? Unless or OC. If there is OC or O minus, the IS cannot bind. Are you guys lost? That's it? Okay. Yep. Review this. Okay. I will do some examples here. I think I will stop. Yeah, this is what happens because when you're doing research and trying to build a network, you get lost because there's this one, that one, and then it's very hard to. Computers do it better now, but yeah. So let me re-explain this. Right, how do I do this? Let's try it. It's gonna be easier, I think. Yep. Yep, I'm gonna show that now. Okay. So imagine 3 scenarios, okay? Let's start by this. Let's start by I plus, just the I plus protein. And then we have the O minus. We have the O plus, okay? And let's put this guy here. Guys, can you look over there? You're good? So, okay. So, OC. Okay. This scenario right here, no lactose, okay? No lactose. Horrible writing, but it's okay. So I plus makes the protein. Because it's O minus, it does not bind here. Okay, so I'll just do an arrow here and do an X on it, right? If there's I plus, O plus, this means that this sequence works properly, so this will be binding here. And if we have I plus and OC, same thing as the O minus, it cannot bind here. Okay? That makes sense? If you have lactose with I plus, the only difference is that it cannot bind here either. Okay? Because lactose will inhibit the I gene, the I protein, so it cannot bind. Okay, this is simple. The hard part is here. So if we have, let's do O minus. OC. So if we have IS, no lactose, IS cannot bind here. And this thing is sensitive. IS can bind here, and IS cannot bind here. Okay? Who has a question up to now? Yeah. No. In terms of DNA sequence, yes, but in terms of effect, no. Yeah. Yep. 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 The only difference is when there is, yep. If we have IS, lactose cannot bind to it. This is the next step. So the only difference is if you have IS and lactose. It does not change. It remains like this. Because lactose has no effect on IS. Because the site where lactose or IPTG bind on IS is different. It's changed. There's a mutation there. And so it does not recognize lactose or IPTG. Okay? Yep. Exactly. It cannot bind to the O minus and OC. This thing here, you have to do a lot of problems to kind of get it. Okay? Yes. Exactly. The IS. The IS. Yeah. Good. Yep. Let me try to make a link there. So the operon is in bacteria, but we do have this enzyme in the body. I think actually the supplements that you take there to lactate, or it's called, or something like that, I think it's just the beta-galactosidase enzyme. So it's the Z gene in a certain form that when you eat it, it gets activated in your body. So yeah, probably that's one of the reasons, the inability to break it down, but also it induces some sort of reaction, so it must have some immune system component to it. Same thing with people who have celiac disease. They cannot break gluten because they're missing an enzyme that breaks gluten. Okay? Cool. Okay. Last example, I'm just not going to draw it, but you can imagine if it's I minus, then all the arrows are X. Because you don't make that. There's no repressor. The I minus. No, when you have the I minus, lactose or not lactose doesn't matter. Good? Yeah, exactly. Okay, let's so let me see how much I have left here. This thing here is becoming just give me 1 second. So we're at slide what? 17. Do we do some problems instead of we do some problems? Okay, and then we continue this stuff next lecture. Okay, let me try to make some scenarios and then I'll ask you questions about this. Okay, let's do this. So I'll be just, yeah. I think I'm giving you the table. I'm giving you the table. Yep. Yeah, I think I'm giving you the table. I remember a plus somewhere there. Yeah. I forgot it's okay. I forgot the question specifically, but I know I'm giving you a table with induction and not induction, and then I think you have to figure out the genotype or something like this. Okay. I forgot the specifics. Okay. Let's do an example. So I'm going to ignore the haploids. We're just going to do diploids so that you can get a sense of trans-acting and cis-acting. And then by doing that, the haploids will become easy. Okay. So let's take bacteria number 1, which has I minus, and then let's see, P, O plus, and then, okay, you know what, we'll do it step by step. In this case here, if I don't have lactose, am I making Z? Okay. If I don't have lactose, am I making Y? No. Okay. What else? If I have lactose, same thing? Right? Okay. Now let's flip it a little bit. Forget the plus, we're going to S. So if I have IS, in this case, am I making Z without lactose? And with lactose? Good. Anyone does not agree? What about the Y? If I don't have lactose? If I have lactose? Good. Good. Okay, let's switch this guy here. So now in this case, if I don't have lactose, am I making Z? Am I making Y? Okay, we got the Y part, right? Okay, so if I have lactose now, am I making Z? Why? Exactly, OC. Because there's OC here, it doesn't matter how strong this guy here, this guy doesn't care, okay? Simple? Excellent. So now, let's do something different here. Let's do, okay. Trick question, guys, ready? What about that? The I, it's true, it doesn't matter, but let's say it's the super strong one. No, RNA polymerase cannot bind here because it's minus. So no matter what you put there, it doesn't care. It's not going to make it, okay? Does that make sense? If the promoter is messed up, there's no transcription, no matter how activated or inhibited it is, okay? So RNA polymerase cannot bind here and go this way. Okay, now let's do, I'm inventing this stuff here. So if we do that like that, do that like this. Jeez, what's happening? Well, apparently it doesn't want to work anymore. Maybe. Okay. So let's say here, let's do I plus, or you know what? Actually, I have a better idea. Still not working this technology, yeah? Jeez. I think even the board is like, this is too hard. Forget. Okay, let's do it again. IS, and then P plus, oh, let me think about it. Minus plus, and then in this case, what's happening? Okay. Yeah. Not good. Try this one. Okay. So, what do I do here? I do I minus, and then P plus, O plus, Z minus Y plus, and there it's O minus. Okay, let's take this example here. Without lactose or without IPTG, what would you make and not make? So if I don't have, so let's go step by step. This guy cannot bind here, right? No. But it can bind here, right? So, and this here does not produce any repressor. So we can imagine this one here does not exist, okay? Just ignore it. IS will come and bind here, but it cannot bind here. And so with or without lactose / IPTG, you will be making Z, okay? Now in the case here, with lactose, are you making Y or no? You're not making Y, because lactose cannot bind to IS and remove it. Yeah. Not in the effect it has on the operon. It's just the difference in sequence in terms of genetics, okay? But in our case, just consider it being the same, okay? Yeah, maybe I shouldn't just imagine this is a C, okay? It's gonna be easier, guys. Sorry? Exactly. 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 Yeah. That's good. Okay. What else can I give you as an example for this stuff here? Huh? Here it's negative, so it's not going to be made from this one. And in this case here, because there's IS here bound, IS bound to the O plus site, even if you have lactose, this will not, the transcription will not happen, because lactose cannot remove the IS from here. If it was, if this here, if this here was I plus, because this one here is I plus, then when there's no lactose, you're not making Y. because the inhibitor is not inhibited in itself. But if you have lactose, it will bind to the I plus and remove it. And so you end up making Y. Okay, yep. The Y minus, just imagine it as the sequence is not there. Yeah. Sorry? Yeah, actually don't, yeah, the result is that, but just imagine it as this guy can travel, okay? This guy can travel. That's pretty much it. This is what I mean by transacting, because even though it's coming from this bacterium, it's still acting on another bacterium's operon. Okay. Let me see here if I can invent some more here. Didn't know. Yeah, kind of. You guys are good. Okay, if I have, what else? I plus IS, that should be simple. Practice this in the tutorial and do some problems on by yourself. You should be pretty good. Okay. I think I'll just continue from here. Yeah, I think I'll continue from here, but I will skip. Okay, we'll do it. It's okay, university students, you can handle it. Okay, so this here is just to show you what I mean by the mutations, okay? So in this case, for example, at the promoter region, if there are certain mutations that will render this, this mutant kind of like a P minus, because it will have severe effects on transcription, okay? So just a single nucleotide change could affect the efficiency at which transcription happens, okay? This is one way of one way through which regulation happens. In this case, for example, you're playing with the Tata box, and if you remember, the Tata box is very important for RNA polymerase and the sigma factor. And so if the sigma factor cannot bind here anymore, or binds but it's very a weak binding, then it will not transcribe as efficiently, okay? So this is kind of the P minus that I'm showing, okay? And then there are other mutations that have just a mild effect, because they do change the sequence, but not to the point where the sigma factor does not bind. It binds just less strongly. And obviously, if you delete this Tata box, for example, the sigma factor cannot bind anymore, and you will have no transcription at all. Yeah. Exactly. Exactly. In the case of the operator, it's a bit similar. There are a few sequences that can turn it into a constitutive operator. These are the modifications. So you can imagine that just one change leads to the operator turning from a O plus to an OC and being always active. Okay. Yep. No. You don't need to remember any sequence by heart. Okay. Just the AUG, which is the start codon, and the 3 stop codons. But even those, I'm putting them on there's the chart there. Okay. It's just the stop codons, the 3 of them, it says stop codon, but the start, it does not say that. It says methionine. So, okay, remember that. And obviously, sequences like poly A tail, right? This is important. Yeah, I don't remember them all by heart there, but there are a few of them, poly A tail, the different codons and whatever. If they were important, I highlighted the sequences in my lectures, okay? The question is, do I mix you up or do I leave it like this? Because there's more regulation on the lac operon. So I think I'll let you practice first. I'll let you get the first part, and then next lecture I will cover this stuff here, because this is a different way of interacting with the lac operon, and this is the positive regulation of lac operon, and they both act together at the same time. Okay, so I'll leave this for another time. Yeah, exactly. Yeah, yeah, with the inhibitor and its ability to inhibit the operon. Okay, if you have any questions, yeah. Sorry. The thing? Well, it's okay. I think we have time for next lecture. It's like a 5-6 slides, but if I add this much, you sounds good. So if you're meeting with me today, we can do it like in few minutes. So we can just go to the office right now and go over it, so that we can all go home a little bit early. Okay? Make sense? See you later.